This is the last video in a series on cellular respiration, and we have already covered the first three steps, and technically there's only three steps, but I like to add in the transition step as its own step. So we've already covered glycolysis and the transition step and the Krebs cycle, or some people call it the citric acid cycle. Either way, this is the last step. And in general, the entire process is called oxidative phosphorylation. And writing and talking at the same time doesn't work here. Phosphorylation. And we'll talk about the name oxidative phosphorylation in just a little bit. It should make sense by the end that we're using oxygen to power the production of ATP, which is what the term phosphorylation is referring to. So just as a review, the formula for cellular respiration that we've been talking about this whole time, C6H12O6 plus 6O2 yields 6CO2 plus 6H2O. We have already accounted for our glucose in glycolysis. We've accounted for all of our carbon dioxide waste. Some of it was lost or released in the transition step, and some was released back in the Krebs cycle. So now we still have to deal with water and oxygen, which are our other two factors in our equation here. So we'll make sure we come back and address those before we're finished. Just as a quick review, though, you need to know that we have been talking about our electron carriers, NAD and FAD. And NAD and FAD are both reduced when they gain electrons in the form of hydrogen atoms. So they look like this, NADH and FADH2. We have gained 10 NADH molecules throughout this whole process so far, and we've gained 2 FADH2 molecules. Now the reason I bring this up now is because I've told you this whole time, oh, we just have formed some NADH and we're going to transport that to the electron transport chain, and I'll tell you about that later. Well, now is the time for me to tell you about that. So just as a reminder, we gained 2 of our NADHs from glycolysis. So 2 from glycolysis. I'm just going to kind of abbreviate here. We got 2 from the transition step. We got 6 of them from the Krebs cycle. Krebs. So just overall, that's where we got those guys from. And then the 2 FADH2s were both formed in the Krebs cycle. Now, interestingly, for each NADH molecule that we bring to the electron transport chain, we're eventually going to be able to produce 3 ATPs. And for each FADH2, we're going to be able to produce 2 ATPs. So that means this one is going to give us 30 ATPs. And this is going to give us 4 ATPs for a grand total of 34. Now, sometimes we get 36 or 38 ATPs, depending on some other stuff that we're not going to go into right now. But just in general, this is an estimate. All right, so let's talk about what all is going on here. First of all, you need to know that this orange area here is what we call the intermembrane space. And this gets really confusing because we talk quickly and we say things like intermembrane space, and it doesn't sound like I'm saying intermembrane space. This is the space between the 2 membranes of the mitochondrion. Now, I'm sorry, I'm not a great artist. Okay, the cristae of the mitochondria, I'm talking about this space right in here that I'm shading between the 2 membranes. That's the orange area here. This membrane is the inner membrane of the mitochondrion. Inner, not inter, inner. The inner membrane. And then over here, this is the mitochondrial matrix. So just to make sure we understand what's happening and where we're located, we are inside the mitochondrion, located on the inner membrane, and we're talking about the intermembrane space. So all of those words get a little bit confusing. So what is happening? This first section here is our electron transport chain. This is where all of these electrons that we've been carrying around with NADH and FADH2 are going to be dropped off. And then these guys will be oxidized back to their original forms, the NAD plus and the FAD plus. And then we'll go back to the other sections of cellular respiration and pick up more electrons, more protons, and bring them back to this place again. And then the other guy over here, this is what's called chemiosmosis. And this is the point, this is like the grand finale. This is where we're going to end up really producing all of our ATP. So all these little gray circles are our hydrogen ions, protons as they were. All right, so if we just start off in the beginning here, this is our NADH. It's brought its hydrogen, which again is just one proton, one electron. It's brought the hydrogen and it's dropping off the electrons. The electrons are going to follow this little yellow line here. They're going to be transported from this protein to this protein to this protein to this protein to this protein and onward. And they're doing this, while they're doing this, we're using that potential energy to build up a hydrogen gradient. So the hydrogen gets split in half. The proton ends up here, and the electron gets dragged along. This first little guy here, we abbreviate with a Q. Its name is ubiquinone. Don't worry about that right now. There's just a Q. And then, again, the electrons are passed back and forth, and this guy's name is cytochrome C. Again, you don't really have to memorize the name, but it would help if you'd heard the word before, cytochrome C. It would help if you had to read it, for example, in a free response question. It would be nice to know the name. All right, so the electrons are being passed from protein to protein to protein, and meanwhile dragging these protons up into the space, the intermembrane space. This guy is representing FADH2, dropping off its electrons, and then of course we remember that oxidizing is losing, right? Oxidizing is losing, reduction is gaining. So right now we are oxidizing. So NADH is oxidized to NAD. I'm kind of running out of room here. FADH2 is oxidized to FAD. And these guys are going to, and it's actually got a little plus here. And I'll explain that at some point. Those are going to go back to the other steps that we've already talked about and pick up more electrons. Now what's happening over here is these electrons are now finished with their jobs, and they've got to be taken care of. We can't just leave all these electrons hanging out. They've got to go somewhere. So what we do is we take some hydrogens, hydrogen ions, and we take half of an oxygen molecule. Remember, oxygen is one of those diatomic molecules. And if you look at this real closely, we take the 2 electrons, 2 hydrogen ions, so that makes 2 whole hydrogens, and half of an oxygen gives us H2O. There's our water, and here it is, our water. And the oxygen is right here. All right, so oxygen is what we call the final electron acceptor. I'm going to abbreviate electron E minus. Acceptor. All right, so what has happened is we've built up this nice gradient of protons up here in the intermembrane space. There's lots of them up here. Think of it as sort of like a hydroelectric dam. There's a dam, there's a lake, there's lots of water, and when we open the floodgates a little bit, the turbines inside the dam can spin and we can make lots of electricity. So this is sort of almost exactly the way this works. We've got a gradient of hydrogen built up. We call it an electrochemical gradient, and that potential energy is going to be harnessed to produce ATP. So as the hydrogen ions are flowing through this molecule here, this protein here is called ATP synthase. ATP synthase, as the hydrogens flow through, ADP plus PI. ADP is going to be phosphorylated. We're going to form ATP. Now, just as a reminder, the whole point of us talking about this in this manner is that we need to understand that we are doing something called energy coupling. We are using a series of exergonic reactions to power an endergonic reaction. The endergonic reaction that we're powering, the endergonic reaction that we're powering is this one right here. The formation of ATP is an endergonic reaction, and if you recall, if I draw this tiny little graph because I'm running out of space, if this represents free energy, our reactants have a lower amount of energy than our product, and because of that we have to invest some energy here. We have to increase the energy, and that energy has to come from somewhere, and in this case it is coming from this hydrogen gradient that we've built up. So we're using all of this series of exergonic reactions that we've been doing this entire time, the breakdown of glucose and glycolysis, all of those many steps in glycolysis, 10 steps and 12 different enzymes doing their work in the Krebs cycle, all of those exergonic reactions. The whole point of all of that is to be able to power chemiosmosis and produce ATP in the end. And that ATP will then be used to do cellular work. All of the jobs of the cell that need energy, that's what we use it for. So I hope you can use this to your advantage and study for the AP exam and any quizzes and tests that you have coming up. And thanks for watching.